going to put a lot of emphasis on is DESeq 2. But before that, I would like to discuss many things which has been already discussed, which can be a little repetition, but still we have to form a basis, we have to form a base, and then we can go ahead and explore DESeq 2 more in detail. So let us begin with the introduction about the RNA-seq. Generally, when we are talking about any DESeq, any differential gene expression, the one thing which comes to our mind is RNA-seq data. But after this high throughput sequencing which has happened and which has taken account into a lot of different application, be it metagenome, exome, you have the differential gene expression, or it's not only restricted or synonymous to just RNA-seq. There are many different different applications which generates our data, and wherein we can find the usage of performing a differential analysis. Be it like in metagenome, we generally do we have a different species, and then we say that dependence at one particular level and one particular environmental condition and at the another. So in this way, you will find a lot of application wherein you will have the usage of this differential expression. Let us go and just focus more on RNA-seq because this will give you a good clarity, and then we can you can also apply this understanding into another application of NGS. A fundamental research problem in many RNA-seq data is the identification of differential expression gene between a distinct sample group. Your sample group can be, you are studying, like yesterday, Dr. Prashant was showing, there was, they were identifying, some gene which are responsible doing of obesity. So similarly, if you have some study in your mind wherein you want to compare between two conditions and want to see, say for example, take a normal example. If you apply a sunscreen lotion on your body, you want to see that what is the change you are getting after application of some chemical. So herein also you are studying a change in what is the expression pattern of the gene change due to some introduction of some lotion on your body. Similarly, you have a condition wherein you compare a tumor and a healthy cell, wherein you want to compare that what are the different genes which are going haywire in a disease or a cancer or a tumor condition. So you will find a lot of differential because of the so much popularity in RNA-seq technology, you have a lot of different method which has been lately come up with performing a differential gene expression. These method can be divided into many different parts. Say for example, some method takes into account of negative binomial model. So you will have EdgeR, which Shalini will take after this session. Then there is a DESeq, then there is BASEC. You have many non-parametric approaches. So you will find many different software for performing differential gene expression. But what software to pick, when to pick, I will address that in later slide. So it entirely depends on your experiment. It entirely depends upon what is the question you are asking, what is your biological question which you want to answer using an RNA-seq technology, and you want to what is that you are comparing right now, whether you are comparing two cells and whether you have replicate in your samples. If you don't have replicate, is your sample having expected to have a large number of the samples which you are comparing, are those going to have a large number of variability, or your sample which you have taken, is it very heterogeneous? Say for example, in case of cancer, we have a lot of heterogeneity over there. So all these things you have to keep in mind when you are choosing a method for your differential gene expression. So right now, what we are focusing here is like when we do an RNA-seq analysis, we do perform a differential gene expression. So we have got many different kind of tools. So my focus here will be today only on DESeq, but I'll touch a little bit on all these which has been mentioned over here. So what do you mean by differential gene expression? Like I said, the differential gene expression is assessment of differential read count of the gene between two or more experimental conditions. So I'll just let you know, about the a basic RNA, RNA-seq experiment. What happens in an RNA-seq experiment, and how do you, say that okay, this particular gene is in high amount. So particular gene. So while you align, you get of reads. So all the game over here is how many reads align to a particular gene. If the number of reads aligning to a particular gene is high, we do say that okay, the expression is high for that particular gene. But we do take care of other biasness, which I will address. The whole game of differential gene expression relies on the count data. And how do we get a count data? We get a count data by aligning our sequencing read to a particular genome, a particular, like if I am studying a human, if I am studying an expression of liver gene and in certain condition at certain point of time. So what I do, I'll take my sequencing, I'll take the sequencing read, and then I'm going to align my reads to the human genome. And while I align the reads, the large number of whatever gene we have, the gene. I'm sorry. So we have the gene which are the number of reads which are aligning to the gene gives a count data. So example over here, you can read it. There can be two sample wherein one sample is a kidney biopsy, the other sample will be a patient from kidney which seems to be perfectly healthy. When we are doing a differential gene expression, we can study like so. All these things come under the study of differential gene expression. So what does a count data actually represent? So like I said, a count data represent a number of sequencing read which is originating from a particular gene. So like we take a RNA from I'm studying an expression of expression profile of a liver tissue. What I'll do, I'll extract the RNA. I'll do the library preparation like we do in NGS. We isolate the RNA, then we chop down those RNA, and then there is a cDNA preparation. After that, we sequence those cDNA, and then we align those read to a genome, like whatever species we are from where we have got this particular RNA sample. And after that, we for each particular when we align it to the genome, it gives us number of sequences which align to a particular gene. So that gives us a count data. So this is a normal basic flow of a RNA-seq experiment. You have your sequence data, and these are the tools wherein when you get the sequence data, you perform some basic quality control assessment. Whatever data you get, you don't straight away start the analysis. There are certain requirement for a particular kind of study. If we are doing an RNA-seq analysis for a human species, then we need certain amount of data. So those things we have to keep in mind. The quality of the data, like yesterday it was explained, we generally go for Fred score. The quality of the data should be greater than equal to Fred score 30. And the if there is an adapter, because in NGS, when we are doing an NGS library preparation, we amplify, these are PCR copies are made, so there will be some adapters as well. So if there is an adapters, you have to trim those adapters. So there is a tool called cut adapt, and there are many other different tools as well. There is a tool called fast p, which does quality control as well as adapter trimming. Then you align your genome, then you align those read to your genome. The genome over here is the species which you are studying. Herein they have used an star, which is an aligner. There are many different aligner, like you must have heard high set, bow tie, top hat. So you have to keep in mind that when you are doing an eukaryotic organism and you are aligning an RNA data to a genome, you have to keep in mind that it should be a splice aware aligner. Then after alignment, you get something called a file format called BAM file. From BAM file, the BAM file has all the information that what are the different genomic location, and how many reads has aligned to that those particular genomic location. And using a tool like high set high set count, or maybe this particular over here they have used a feature count, wherein they extract those they extract the number of reads aligned to a particular genomic location, and in this way we get the count data. So herein, when we get the count data, the next step will be identifying the differential expression gene. So over here, you can have, like we were discussing in the previous slide, that for differential expression, you can choose any number of tools, because you have lot more tool over here. You can use EdgeR, DESeq. So it entirely depends upon, while choosing. I have kept one slide wherein they have compared, like what tool to use when. So herein you have to see your experiment, and then you have to decide accordingly that which tool you have to use for your analysis. So differential expression steps are followed in this particular step. What we do, the count, how does a differential expression analysis takes place? Till generating a count data is okay, fine. Once you get the count data, a count data will be what you will have. Think about a matrix over where you have the rows, you have the column. In the column you have the name of the gene, and the rows you have the sample, and then the entire matrix has how many reads has aligned to that particular gene. So after you get the count data, it is not that you straight away start with the analysis. There are many different steps which you have to keep and look into your data, that whether your data is good to go for a differential expression or not. So the two steps which we call are very important for any differential gene expression analysis is, first you have to see your data for normalization, how normalization has to be taken care. Second is that you have to look that, okay, how is my data, whether my data is good to perform a particular kind of differential expression or not. So for any differential expression analysis, you have to keep in mind the two steps, the normalization and the QC control step. In normalization, I mean two things, because you understand this particular term, that the expression of a particular gene is said on the basis of how many reads align to that particular gene. So there can be, it can happen that we have a two sample over here. One is say in this particular picture you can see that you have sample A and sample B. So it can be, you have sequenced, you have two sample. In sample B, you have sequenced a less amount of data. And in sample A, you have sequenced a large amount of data. Say, for example, sample A has 20 million data when you have started with the analysis, and sample B has 10 millions data. So herein you can see that the amount of data for sample A and sample B are it is very it is 50% less in sample B. So obvious, if a particular gene, say for example gene X, is present in sample A, due to its having a large amount of sequencing data which has been generated during NGS library preparation, large amount of data, a large amount of data will lead to that particular gene. If I am comparing a two gene in both the sample A and B, so obvious the due to large amount of data present in sample A, it will have lots of read aligning to that particular gene. So this when we are performing a differential gene expression, wherein we are going to say that okay, what is the expression of a particular gene in comparison to another sample. When we are comparing, say for example, we are comparing between healthy and a tumor cell, and if we start with large amount of data in the healthy cell compared to a tumor cell, obviously particular gene will have a large amount of data aligning. So we have to keep that also into mind. So the first step over here is normalizing this particular biasness, so that the gene which we are comparing, we show that okay, this is the true particular differentially expressed, not because of the large amount of data which has been generated, and there is a disparity in the amount of data between the two samples. So this particular kind of normalization which is done via any you have lot of tool for differential gene expression. Some of the tool do it inbuilt, and they have their different method. So you must have read in if you have come across RNA-seq data, you must have read the people tell in terms of FPKM, RPKM, the expression. If someone asks what is the expression value of that particular gene, we tell tell in terms of FPKM or RPKM or TPM. So what are these? These FPKM, RPKM, TPMs are the normalization method, and these normalization method does this only, that it removes the disparity if it is due to sequencing depth. If a sample is having a large amount of data and another sample is having a lesser amount of data, this should not give a false expression. It should not give a kind of expression which is not true for this particular sample which we are comparing. Second is your sequencing length. Say for example, if anything, as we know, if the expression in RNA is always about how many reads align to a particular gene. So if we are comparing within the sample, because this sequencing length comes within the sample. If a gene, we are saying that, say for example, we are saying in liver cell P53 is expressing in a larger amount compared to AC2. So we have to also take care that if we are saying these things, the expression, the gene length has to be taken care of, because if a particular gene is having a longer length, for sure it will have a large amount of data aligning to it. So that also we have to take into care. So this sequencing depth and sequencing length is well taken care of by very popular normalization method like RPKM, TPM, and FPKM. So so this comes under the normalization, that you have to understand that before performing any DE expression analysis, you have to normalize your data. Second comes the quality control. If we are comparing if we are having a replicates and we are comparing in quality control, we have two different steps. One is sample level quality control, another is gene level quality control. So while performing any differential gene expression analysis, we have to see that if any gene is either not expressing any, if it is not having a very less expression, like it is having no expression, or only expressing in one of the sample, that things we have to remove, because that is not going to be any useful for differential gene expression. When we are saying something is differentially expressed, we have to have that particular gene expressed in both the condition. If I am comparing, there has to be someone to compare with. So so those kind of gene which is having only sample specific kind of expression, those are also removed removed before the DE analysis, differential gene expression analysis. If any sample is having a very outlier kind of expression, that is it is not falling into the it is very outlier, that has to be also removed during differential gene expression. And then comes the sample level quality control. In sample level quality control, if we have replicates in our sample, like biological or technical replicates, we have to take care of that. These replicates are falling into showing a correlation, and these can be done via plotting a PCA plot, or a or a clustering over via clustering. We see that if the correlation, if two samples are well correlated, at least it should have a correlation value between 1 to 0.9. It should have it should be correlated. We can expect 10% values getting here and there, but at least it should be 90% correlated. So to sum up what I have discussed till now is like we have a RNA-seq data. With the RNA-seq data, we generate a count data, and count data are generally used. A count data is the only your go for any differential gene expression analysis. So while you are performing any differential gene expression analysis, you have to take care of two things in your mind, that normalization and your sample QC. Those things you have to keep in mind. So with the DESeq, why I prefer DESeq 2 first is that these particular things which we have discussed right now, the sample level QC, gene level QC, and normalization method which has been used by DESeq, it gives a good result in even in the terms. If you have a very small set data, DESeq is always very reliable, and it gives a less false positive. So also, when you're performing anything with DESeq, you don't have to worry about all these things. These steps are done by DESeq itself. But when you are doing EdgeR, then I think you have to perform these steps prior to doing a differential gene expression. So these are the different software packages for detecting a differential expression. You have EdgeR, you have DESeq basis. So these are the different normalization method which has been which is used by these software. Normalization over here means the taking care of these two things, sequencing depth, sequencing length, and there is also called sequencing composition. Those things you have to take care. So these are the different normalization method used by these different software. And these are the read count distribution and differential expression test. What test it performs for calling anything significantly upregulated or downregulated. So what point you have to keep in mind when choosing a differential expression analysis software? So when you have a, like I said, when you have a when the number of replicate is very small, you have two condition. One is healthy, another is tumor, and you have only 1H or 2H replicates. Then you have to be very cautious while picking any differential analysis tool. But if your sample is larger, then you have to not much worry. But if you have a number of replicates is small, then you have to carefully pick the tool. So Lima and DESeq seems to be the safest choice in those conditions, and there has been many publications wherein these two particular differential expression tools gives up quite good result. But if you have a large samples and you have a large number of replicates, you don't have to worry much. You have a large scope of choosing either many. You can even, I'm not saying that Lima and DESeq are not good for a larger subset. It is good for that also. So, but when you are doing with a smaller set, you have to take care, what tool you are picking. With the larger set, you are good to go with a large number of choice. You can pick any of these tools. And When the number of replicates are relatively large, you have the non-parametric methods are as publications are there that that proves that this is a large number of, it is very useful. So in non-parametric you can see what all noisec, you have samsec, so these can be your good to go tools. Also, yeah, also I told earlier also that it entirely it also depends upon what is your experiment, what is your biological question which you want to ask via this particular differential analysis step. So you have to take care whether if you are if your sample, if your data is showing a large number of heterogeneity, then you have to be you have to take care of choose a tool which will address this in a good way. So here also DESeq and Vima are the safe choice. So you can see, I am not voting for DESeq, but yeah, it does give, publications are also there which support that it gives a better result compared to other tools. But it entirely depends upon your experiment. So you have to publications are there, where they have compared all these tools, but there is not, the results are not this overwhelming, saying that okay, pick this tool for particular for your differential expression analysis. Those studies also says that you have to take care of many other things in your mind, like your data, your sample, your replicates, your heterogeneity, the how many samples you have. So all these things you have to keep in mind while choosing any particular differential analysis tool. Rest there is no such any data available which says okay, go for this for all of your answer. Differential expression analysis with DESeq. Like said earlier, that DESeq uses a non-binomial model for performing a statistical test for differential expression genes. So we do know that it performs from this table. We can see that DESeq uses a DESeq size factor normalization method, which is also a mean geometrical normalization method used to normalize the data. The read count data assumption is made on negative binomial distribution, and the differential expression analysis which it test which is performed is exact test. So the test is actually to tell that okay, whatever is we are saying that this particular gene is expressing in higher it's upregulated compared to the other sample. It has to be significantly valid. It cannot be like just out of fluke you are getting this. So that's why we perform a test, to test our hypothesis that okay, this particular gene is in true sense upregulated in comparison to other. So yeah, so the steps performed by DESeq functions are the estimation. So when we do a demo session wherein we are going to you utilize the DESeq tool, you just have to perform only 2 steps and your work is done. So you don't have to do much preparation of data, this and that, when you are doing a DESeq. And yeah, we are talking about DESeq and we are saying that DESeq is this and that. So what are the steps which DESeq perform? First is that it estimates the size factor.
Actually,
Can
I'm
you
not
see
able to see
over here? Oh,
screenshot.
sorry. So there is no glitch in the program. It's just copying from here and then copying, pasting over there. That's why there is a problem. If I copy direct from the HTML which I have created earlier, so it is working fine. So I'll just come here, and here was I was talking about. Look over here.
Yeah.
If you can see that this is a complex design wherein I want to compare the group between the sexes. I mean, I want to compare male, female, and I also want to compare between the age, and I want to compare the treatment as well. So it will be male, age,
Okay.
13, 11, 13. So you can see, so this is a
Okay.
complex kind of design. So if you want to,
Okay.
like, I want to compare, like I gave you an example, I want to compare the healthy group, male, female, male, female, to the tumor group, male and female. So what I'm going to write is like, so in the design I will have male, female. So I will write here, there will be header for that, no, sex. So I'm going to write a
Okay.
sex, and then I'm going to write a group wherein that will be, tumor condition. So it will be
Okay.
like this, sex and plus condition. So this is,
Okay.
I'm saying that compare the male and female and the condition as well. Take care of their condition, that they are healthy, or they are what do you call, the tumor patient or not. Fine.
Okay.
So
Okay.
yeah.
So if I understood correctly, in the PDF which was given, so
Uh-huh.
the third column liter will be ignored, right? For design purpose.
Yeah, the third column width is ignored. I should not have added, but I only kept it just cause, because sometime if you want to, it can be, that you have your matrix is ready, and you don't want any two column to be compared. So herein you can write the width, that you don't want case 5 and case control 5 to be compared. So that you can do. So what it will do, that it will have your data, but it won't have case 5 and control 5. So width is that. If you don't want to include that, just delete this particular portion and close the bracket. It will be done.
Okay. Okay. That's
If
great.
you don't want to go into that phase, just do that. Fine. So
Okay.
here in what we
Sure.
were doing, first is like PCA. This is done. We have generated the P plot, and then we are doing a PCA plot. So this is it. I was writing this. Fine. Good. Just and then you have PCA. So your PCA is generated. So your PCA plot is saying that your data is actually the data set which you are having. All the replicate in the control are falling into the cluster. How is the variability in both the group, the control and the case? Fine. So in this way, what are these? Why we are doing this? Actually, DESeq is done. We have performed the DESeq. These are only a way to represent our data. If you need any up-regulated and down-regulated, the REST file, it has everything. The REST file has. If you have come across with the result of differential analysis, in the first column you have gene name, then you have some expression value, log to fold change, and then p value, p adjusted value. So this is what it is there. So on the, if it is significant 0.05 and the log to fold change is greater than equal to 2, then we call it as a upregulated. If the log to fold change with the significant value is less than -2, equal to or less than -2, so then we call it as a down regulated. So that is done. These are just the representation of your data. Fine. So PCA is done. We are doing a volcano plot. So this library enhance we have already done that. We are calling our we are calling this particular package. I think I have taken a lot more time and
Yeah, once they start reading exercises, probably there will be some questions rolling, Ankita. Yeah.
yeah, I do know. Yeah, so I'll just run fast on this. So no, I'm not doing this. So yeah, so enhance volcano plot, you have here copy. So you have your enhanced volcano plot generated. So this is the same formula. It's just over here the and after that when we are done with it, so you can read over here, you know, what is the volcano plot and all that, because I have explained it in very simple term over here. So the upregulated genes are generally into this particular range, and then in the right hand side, and then you have the left hand side, and these black ones over here are the baseline, which is not showing any expression, but any significant change between the two conditions. So all these things you can find over there. And then comes, you know, very much important picture of differential gene expression, that is heat map. Many of my client always ask for heat map all the time. So to make a heat map, we do need certain packages. So packages are already there, so we are just going to call it here, because right now we need those packages. So we need deploy. I'm going to call it. I'll just, then I need this table, and so all these packages are done. So herein I am just writing res up. So res up is what? Res up will have all the upregulated gene data. So if you look at this particular formula command over here, it is saying that take the res file. Res file has our differential gene expression analysis result. If the change in as data frame rows column num, if the change in the gene is log to fold change is greater than equal to 2, upregulated. So you have to write all those things. If the log to fold change of that particular gene is greater than equal to 2, then write it into the rest app. So we are going to just copy this and paste it over here. And if I'm going to head rest up, sorry, rest up. So yeah, so these are the genes which are upregulated with a significant change. Okay, so it has written also, because I am mutate over here, so it is writing over here that these are upregulated genes. So similar way for downregulated, like I told you, for downregulated the expression value and fold change is fold change is what fold change is equal to less than equal to -2. The similar way it will give us a list of. So generally when we make a heat map, why I'm doing it, when we make a heat map, we make from upregulated and downregulated gene only. So that's why from our result, I am extracting only those genes which are either upregulated or downregulated with a significant change. So upregulated I have done. Now the same formula goes for downregulated. Only change is the log to fold change is less than equal to -2. So if I do head and I will write res down, rest down, it is going to give me the down regulated gene. Fine. After that, what I have to do, because at this I am preparing for differential gene expression. So I have my up regulated and down regulated gene ready. I am going to combine both of these and then generate a heat map. So over here rest down and all that. So now I am over here I am just binding. If you can see that rest flit has r bind. R bind means that I am just adding all these two into a single file. Fine. So rest filter has the data which will be taken for differential gene for making a heat map. Heat map we generally make with generally what heat map we only make with up and down regulated only. So now this is like differential gene expression heat map plot. For heat map, what function we are going to use for DESeq is BST. Like we used different for M plot and all that. For different for heat map we will use a package from DC, that is BST. Fine. Okay, and then we will take it. This is the library, because P heat map is what give kind of data. Now, so this is going to take our VSD count data and make a heat map for us. And no, I'll just okay. Okay, just give me 1 minute. So if you get an error, these errors are very simple. If you read the error itself, it will tell you, that I forgot to write this particular name in that particular file. So just so this is what your heat map is. So in this way, you generate a heat map. So these are just the basic, of differential gene expression. There are many different packages which you can use. So once you perform this with you in your, or maybe R cloud, or maybe you can, this R studio work on any operating system. We generally, actually, majorly, I use Unix operating system, but this also works on Windows and Mac. So if you can install it and you can Google, I mean, just go through this particular document, you have everything. I ran in front of you using all this command. So if you do once with yourself sitting at home, you will get a lot of confidence. Second, I am always available on this particular email ID. You can always write to me or if it is not working, ma'am, there is a problem. I'm always will be happy to help you and give you my suggestions or input. So yeah, please, if anyone is having any question in this entire session, can tell. Okay, hi Ankita. Okay. Yes, Shalini. The DESeq 2 doesn't work for the replicates. You have to use DESeq. Not DESeq 2 doesn't work for replicate. You have to use DESeq. So but even the DESeq itself, DESeq 2 people, the community itself said that you should not perform an RNA-seq data without replicates. So they also highly discourage, but still you have a savior at your end, that is DESeq. DESeq 2 doesn't use this.
So Ankita,
Thank you.
I
Thank you so much.
I'll just like to add here that see, definitely for RNA-seq analysis, best practices doesn't suggest to go without replicates, but if you are doing some pilot experiment and you just want to see how your samples are behaving, and you want to see how whether this will work or not, then in that case you can use your samples for your pilot experiments without replicates. But definitely for sure, nowadays any best practices, if you see for RNA-seq, they won't recommend using samples without replicates.
Okay, so I guess nobody is having any query. Either you followed it all or not at all. So I assume the first, that you followed me.
Hello, ma'am.
It's been very much useful, Ankita. Thank you so much.
Thanks.
So I think it's time the participants made the exercises. I think it is very important. So please do the exercises. I think Ankita will be there for the next few minutes for lunch. So please feel free to ask any questions once you do the exercises. Thank you so much.
Hello, Ankita, ma'am.
Yes, please.
Okay, on the final heat map, right? And we have just a couple of inference points, like from the overall exercise, how we are finally analyzing the heat map. based on the exercise we just
Okay,
had.
this one you are talking about.
Yeah.
Uh, Yeah, so this is like, this is taking a two-way clustering. First, over here is all the gene which is clustered. So gene which is showing a similar kind of expression, they are clustered in a similar pattern. And then you have over here is your the samples, the case and control. Fine. So
Okay.
this is actually, generally when we do, we get a very good heat map if the data, the up and down regulated genes are, low. If you have somewhere around 100, you will get a very good kind of plot. But with
Okay.
this was a demo data and all, and we just wanted to, so because of that, this is showing a lot of cluster. So over here you can see, the pattern is like that. These particular genes are, this is, I cannot magnify as well, so yeah, I can. So you can see over here, these are falling into a clustered. So
Okay.
if you have to, when the data is low, you have all the names as well for the genes. So
Okay.
then you can easily say, okay, say for example, there was a P53 gene. Okay, it was showing this kind of expression. It was a dark green, and then it is getting a light green, and then it is turning into red. So in this way, you
Okay.
can easily say. But this being a
Okay.
very huge, you cannot say. You cannot say the same thing.
Okay.
Yeah.
Actually, if I understood correctly, I am looking for contrasting colors, right?
Yes, you are looking for constructing the. So
Okay.
okay, so I may have missed the green color shows that it is down regulated. So
Okay.
down regulated, this means that the expression value is a log 2 fold change between our two condition which we have compared is less than equal to -2. Fine. And if it
Okay.
is a red color, the log 2 fold change between the two sample is greater than equal to 2. So that's why it is red. Fine. So over here you can see this particular legend which is showing you the gradation of color. So if it is this green, it means that it is below the 2 also, and if it is dark, very dark, then it is greater than 1. And whatever
Okay.
is between these conditions, this says that there is no significant change. It is same between the two conditions.
Okay. Okay.
So
Right.
just
Thank
because you don't have this expression, I mean less amount, you could have the name as well. So that makes a lot of sense.
you. Okay. Understood. Thank you very much.
Yes, Dr. Ashok, the only problem with the DESeq and right now what happened, they have launched R Cloud. With R Cloud, they have this version. If you print, the version will come 4. And the bioconductor package which we are using DESeq is still not updated. So that's why you will have this particular option problem. So if you are so much interested, I would suggest that if you have system, you just
Yeah,
download.
I'm struggling for the last
Yeah,
half
you
an
don't
hour.
don't struggle. I'll help you.
No
Maybe
issue, no
I'll
issue.
help you with this. I can do.
No, I understand that.
No, no, no.
There's a
I
compatibility
want
issue.
you.
No issue.
I want you to run this because you are very much interested. You can do one thing, that you install a R Studio 3.6. If you cannot do, you give me a remote access. I'll do for your system.
No. I will do that. No problem. I was
Yeah,
just
and
interested in installing our studio on cloud. That's why I'm
yeah.
interested.
No, no,
No issue. I understand
that
that.
you
There is
can
a
do
basic issue.
that this particular thing will work if you give some time, because they have right now introduced this
Yeah, I understand. I
version.
understand that.
So
I
that's why it
understand.
is having some issues.
Yeah,
That's why.
yeah, I really appreciate the open source technology, and we have a patience to get this updated, and we will be using no issue.
Yeah, yeah. Okay,
Thank you very much.
thank you. So yeah, please go through the document if any case, and even if you have my email ID, you can always write to me about anything, any kind of query if you have.
Yeah, can you share your email ID on a chat box
Yeah,
so we
I
can?
have shared my email ID. I'll give you.
Thank you. That was a nice session, and the way you conducted the session is very smoothly and very easy to understand.
Thank you
But
so much.
so it's our bad luck we are not able to follow in hands-on
No, no,
session.
I mean, actually the things are little more
Yeah,
clear.
I understand.
They might say that online works always, but when you sit and have a offline session, one to one, then
Yeah,
it is
I
it
understand.
has its own charm.
Yeah, I understand. Thank you, doc. Thank you very much.
Thank you. So I've shared my email ID and the phone number. In case of any query or any confusion, you can always, it's not that, for sake of saying, I would love to help, and I would be very happy seeing that you people are replicating the things which has been shared. So
Thank you very much, ma'am.
So, are you going to wait for some time, or is the session is closed, or what?
yeah, so I guess I think my session is done.
Yeah.
In case, Dr.
Just
Prashant, let me know. I mean,
Yeah. Thank you, Ankita. I was just about to ping you. I just thought, maybe until
Galaxy.
1 o'clock at least, that they should do exercises. So if you can be here, for few more minutes, if they have any questions, they'll ask you. Okay. Yeah,
Yeah, I am
please.
there. I am there. I mean, yeah.
Yeah. Thank you so much. Shalini, probably you can take, as per the schedule, maybe at some point of time, maybe after they finish the exercises.
Yeah, sure.
Okay. Yeah. I am Shalini and Ankita. So please go through the participants, exercises. So if they have any questions,
Yeah.
I think, and then probably at about 3 o'clock, I think Shalini can give a kind of an EdgeR demo. Is that fine, Shalini? Yeah.
Yeah, sure.
Yeah, please. Huh? Yeah. I think this room is still open like this, but we encourage all of you to do the exercises. So be it the Galaxy exercises or what Ankita has wonderfully explained through the art studio. So please do this, and most importantly, please come up with some kind of brainstorming questions. So thank you so much.
So what I'll do, I'll share the R Studio installation is very easy. So I'll share the link, and if you want, you can install that as well. So I'll just if anyone is interested in whatever they are doing, I'll just